All right, so let's go ahead and get started. Yes, we are starting with males because they're easy and we get them out of the way.
2 minutes and you're done.
30 seconds. You guys are off the hook today. All right.
We're slowly losing our minds.
It's clear. All right, we're gonna go into the males. We have 90 slides. Thus far, I've never gotten through all of it in just one lecture. So whatever we don't, I will record and post it on Canvas for you guys. And then Friday we'll start with blood. Okay. So a lot of this is anatomy at the beginning because again, you have to talk about the anatomy to understand the physiology a little bit, and we actually call that more functional anatomy. So we'll go through those slides pretty quickly. So first, we'll start with fetal development again at about 7 weeks of gestation.
Sorry, can you turn the speaker off?
That's the fan.
Or like the volume? No.
Oh, is it the speaker?
Yeah. Sorry. Oh, yeah.
Better? All right.
Sorry.
All right, so we'll get started. While you're still an embryo, there's a gene on the Y chromosome. All right, so it's not just a broken X because females don't have this gene on their other X chromosome. It's called the SRY gene, determines sex. That gene gets turned on, it starts expressing protein called the SRY protein. What that does is towards the end of that 8 weeks embryonic stage, it's going to make a gonadal ridge, which we'll see on the next slide. And that gonadal ridge will then start secreting testosterone. And then human chorionic gonadotropin from the placenta will further stimulate what's going to become the testes to continue secreting testosterone, and that's going to drive male genitalia, distinguishing from female genitalia. So starts with the turning on of this gene, continues with the production of testosterone. All right, and then at about 30 weeks of gestation, the testes, who are up here where the ovaries are, descend down into the scrotal sac and become, for all intents and purposes, functional. Okay, so you're going to see this again in the females lecture. So this is what we look like. Can't distinguish a male from a female embryonically. Then as soon as that SRY gene gets turned on and testosterone starts kicking in, then you start going down this developmental pathway. And then pretty early on, a couple weeks into being a fetus, you can tell whether or not it's a male or a female. So by the time you get down here, it's week 12. So about 4 weeks to do that. All right. Then, as we said, about 30 weeks, testes descend. There is a structure in the abdominal cavity called the, I love this, gubernaculum. It's fun. All right, what that does is it guides the testes down into their final resting place. Makes it sound like they're dying. So they guide it down through the inguinal canal and put it in the testes and then remain as a remnant structure on the floor of the testes, anchoring the testes in place within the scrotal sac. Okay? It's not made of muscle. It's connective tissue. So what it does is it kind of slowly degrades. The connective tissue slowly degrades, and that's what's pulling the testes downward. If that doesn't happen, you end up with a condition called cryptorchidism, which means the testes fail to descend and you're rendered infertile. Now, why is it important that the testes remain outside the body? Temperature, what about temperature?
It's too hot in the body.
Right. It's too hot in the body, 98.6 or whatever our body temperature is supposed to be now. It's too hot for sperm development, so they need to be outside the body in order to do that. They fail to descend, they maintain that internal body temperature, and you don't get proper sperm development. All right. If during fetal life, so the way we're looking at this, why did I do that? Come on. If during fetal life the testes fail to descend, not only are you going to have trouble producing sperm, but you're going to not develop fully into a male. You're going to develop female sex characteristics because the testes all around aren't functioning properly. It's not just that they're unable to make sperm. Testosterone production starts failing as well. If we lose our testes before puberty, so they descend, they're working fine, you climb a fence and get caught, they come off. Or if you're back in the Middle Ages, they just whack them off. Did that happen? You guys ever seen Game of Thrones? Okay. So before puberty, then you develop infantile sex characteristics. So your body's going to develop as, like, think of an infant child. Like, they have the bigger bellies, they're like fat deposition, and very underdeveloped genitalia. That's what a male will look like. Taller, reduced muscle mass, etc. And if it's post-puberty, if you get rid of the testes post-puberty, you might have some regression of secondary sexual characteristics, but a lot of it's going to be related to loss of bone and musculature because of the loss of what? The big T.
So if they fail to descend and you have them surgically descended, then can you start producing proper, like, sperm properly, or is it no, like, the process is done?
I'll give you the answer that you don't want to hear.
Okay.
It depends.
All right.
It depends on how far gone they are. All right, so this is kind of an overview of the male reproductive system, the different parts. And these arrows are coming from specific glands and what they produce. We're going to go through all of this stuff, but this is another one of those good summary slides to come back to in case you're like, all right, I just want a quick view. What do these things do? All right, we'll begin with the testes. All right, they are the primary male reproductive organ. I want you to distinguish a reproductive organ from a sex organ. They are different. These are designed for reproduction. Penis designed for sex and reproduction. All right, they're ovoid-like structures, very similar to, like we already described, the ovaries, located in a scrotum, which is a sac-like appendage hanging outside the body that, again, is involved in temperature regulation. Two jobs: they make sperm cells and they produce sex hormones. So, again, if they don't work, it's not just that you're infertile, it's that your secondary sex characteristics all start to go whack too. All right, they are suspended by something called the spermatic cord. Now, what you see right here is the ductus deferens or vas deferens. Okay, what happens if you cut that in a male? Surgically. You can't have kids. That's a vasectomy. Okay. It doesn't hurt, but there's a lot of pressure associated with it. I can't have kids.
It's reversible too.
Do what?
Isn't it reversible?
It is reversible.
Or is that different?
Nope, it's reversible. Sometimes it reverses itself. So when I got my vasectomy, one of the things they told me is, all right, make sure you bring in a semen sample, so we can test to see whether or not it took. I said, I'm good. I work in a college. I have access to microscopes. I'll check myself.
Is it him?
No. He was fully planned. I'll tell you about my daughter another time. She was planned too, but there were some complications there. She is fully aware of those complications, so I'm not saying anything she hasn't heard before. Anyway, so you have this vas efferens, but it's in the spermatic cord. So the spermatic cord also has blood vessels, nerves, and connective tissue. It's a bundle going down to the testes because those testes have to have a blood supply. They have to have a nerve supply. When it gets cold, there's muscles that contract and go whoop, bring them back up towards the body. So we have to have things that regulate that. So they come down, those structures come down with the testes. All right. As you saw in the anatomage table, those testes are encased in a dense connective tissue called the tunica albuginea. All right, and you also have a tunica albuginea surrounding the ovaries. We'll talk about that later. And then what that coating does is it infolds and separates the testes into compartments. All right, and those compartments, or where it separates, that's called the mediastinum testis. All right, so a little wall between the structures of the testes that will divide them into lobules, and those lobules contain the structures called seminiferous tubules where sperm are made. Okay, so connective tissue surrounding, folds in, separates into rooms. Those rooms are filled with coiled tubes that make sperm. I always worry that I'm not recording. All right, more about this. Seminiferous tubules. What you're looking at here, if we take one of these tubes, turn it on its side and do a cross section and look into it, this is what you're seeing. All right, this right here would be the lumen, so the inside, and then from here to here, you will have the structures dedicated towards making sperm. All right, and when sperm are made, they start in the outer periphery and work their way toward the lumen. Okay, they start at the periphery and work their way toward the lumen. Between each one of these seminiferous tubules, you notice we have a bunch of smaller cells. Those are called interstitial cells. Those are the cells that make testosterone. All right, so seminiferous tubules, strictly sperm. The interstitial cells are what produce the testosterone. All right, and here's just a blow-up of that. These would be like stem cells that go to make the sperm, and here would be the almost fully mature sperm as it reaches the lumen. All right. As the sperm get formed and they make it into the lumen, they all go into this, they all converge in these tubes into this area here called the, is it on here? It's called the ready testis. Okay. Was it in the previous slide?
Right there, in the bottom right.
Yes.
The tubules form.
Right here. Ready testis. You've heard this term. It's used a lot in biology, especially in tissues. Reticulum. It's like a network of tubes, okay, where they're all converging. And then they all then go to this other structure sitting on the outside, which you felt in those fake testes that we passed around in lab, called the epididymis. What's plural for epididymis? Epididymi. Okay, when cells enter the epididymis, they are immature, which means they're not capable of swimming. All right, a couple distinctions in words here. When they enter the epididymis, they're not capable of swimming. In the epididymis, they mature and become capable of swimming. Does that mean they are swimming? No, it means they're capable of. All right, so they undergo changes within the cells that make them capable of doing the swimming thing, and they don't actually start swimming until they get pulled into the ejaculatory process. All right, so this epididymis goes downward, coils, and then starts going upwards, and eventually becomes the vas deferens or ductus deferens. All right, so what does the epididymis do to mature? It secretes glycogen. Remember, glycogen is just a large storage source of glucose, as well as some other growth factors and hormones that support sperm cells and help them mature. To get from this point to this point takes about 2 weeks, 1 to 2 weeks. All right, vas deferens then, thin muscular tube, starts at the epididymis, goes up, around the urinary bladder, back down until it reaches the ejaculatory duct. All right, so this long tube that goes up, around, and reaches the ejaculatory duct. It's muscular, so what does that mean it does? Contracts. And it's lined with pseudostratified epithelium. So it's got a thick layer, and it contracts. What it actually does is, since they aren't capable of swimming, they don't swim their way up the vas deferens. You have muscular waves of contractions that push them up. What are those contractions called? Peristalsis. So peristalsis does not just exist here. It exists in other structures in your body as well, such as the vas deferens. Who's asking?
Do they have like their own pacemaker cells then? Or what mechanism causes the peristalsis?
That's a really good question. I don't know the answer to that. No. If I had to guess, this is just based on the way things are set up in it, it's probably a continuous peristalsis stimulated by the parasympathetic nervous system because sperm are constantly being formed, so you're constantly trying to get them up to where they need to go. But I will look that up. All right, seminal vesicles. So we've got our vas deferens. It's coming down here into the ejaculatory duct. And then you have these two little structures in the back of the bladder. So notice they're like ears coming off the bladder, called seminal vesicles. Seminal because they are what start secreting the things that, the substance that makes up semen. Okay, and again, you ejaculate semen, not sperm. Sperm just happens to be a cool byproduct that gets people pregnant. I don't have that superpower anymore. So, what is in that fluid? It's alkaline. So is the pH high or low?
High.
It's high. Why does it need to be high?
You don't protein can be measured.
No?
Vagina's
Good guess.
acidic.
The vagina is acidic. We had an instructor here. He taught A&P, really tall Boston guy, tons of energy. And he's like, all right, why does it need to have a high pH? Because you get that vagina is a hostile environment. It's hostile, so they need to protect themselves. It is. It is true. I mean, never mind. Okay.
Speak your mind.
No. So pH regulation, and it increases sperm fluid volume. Because remember, you don't want to ejaculate. You want it to be in a fluid. All right. It also contains fructose. All right. Fructose is a sugar, and so that is the energy source for the sperm on their journey into the female reproductive tract. Like they pack their what do they call it? Trail mix. It's their trail mix, and they go on an adventure. All right. See?
Like the good kind with M&M's.
Do what?
The good kind, like with M&M's and stuff.
I mean, it does. It's fructose, so it's a little bit sweet. Yeah, not salt and vinegar almonds. Also contains prostaglandins. All right, what prostaglandins do is they stimulate smooth muscle contraction in the female reproductive tract. And that smooth muscle contraction, once the sperm are in there, helps them move towards their goal, which is the uterus. All right, prostate gland. So that comes after the ejaculatory duct and then eventually becomes part of the urethra. So it's on either side right here. And remember, we talked about if you stick your finger in the rectum right here and feel forward, you can do a prostate examination. So to determine if it's getting large, prostate cancer checks. A lot of time they use PSA now, but there are different PSA tests. I looked a little bit this morning. There's some controversy on whether or not PSA or digital rectal exam is the gold standard. So I would say it's still up in the air. I don't have the answer to your specific question. I didn't look that up.
That's okay.
So what the prostate does, secretes a thin, milky, again, alkaline fluid that the whole purpose is to increase volume. All right, so you want an increased volume so you maximize the potential to get it into the female reproductive tract. Now, this fluid is going to then enhance the motility of sperm. So from the prostate is where you really start getting the sperm starting to be motile. And from the time the sperm enter the prostate to the time they leave the body, not a very long time, right? So it's a very effective means of producing mobility. All right, then the bulbourethral glands. These are these two little glands right here, just distal to the prostate and on either side. What they do is they secrete mucus, a mucus substance, and often that is the first thing to be secreted outside the urethra. So what they usually call that is the pre-ejaculate, and that's what lubricates the glans penis for insertion into the vagina. But if you've ever had sex with a female, it's usually the female that provides most of the lubrication. All right. Scrotum. Again, that's a sac that encases the testes. Specific things about the scrotum. There is a septum within the scrotum that separates the two testes on either side. So no matter how much you try, you can't like switch left and right. Like you can move them around, but you can't like, there's a wall dividing them. So they knock on the wall and say hi to each other every once in a while. Surrounding, just underneath the skin of the scrotum, you have something called, it's a super thin layer of muscle called the dartos muscle. And that muscle is the thing that contracts in response to cold. So if you've ever seen testes that are just kind of like really hanging there, they're hot, they're trying to get away from the body, this dartos muscle, when it gets cold, they go, and it looks like literally a walnut. Like it gets all wrinkled. It has what looks like rugae, which means that it can stretch back out. Okay. We also have a cremaster muscle. That is the muscle that's coming down here. That is skeletal muscle. It's voluntary, so you can contract that and pull them up yourselves too. I made a joke. I used to make a joke in my undergraduate class. That's not appropriate anymore. But I would say, just in case, like a little kid comes running after you under your leg, and you can go whoop, and they run. Only before it wasn't a little kid, it was a little person. So that's not appropriate anymore. I've matured. All right, penis. Cylindrical organ whose job is to convey urine and semen, as well as provide a method to distribute those things to the proper places. It's specialized to undergo erection. We'll talk about how that works later on for intercourse. And what you see on the right here are a bunch. Somebody let me borrow a book years ago that had a bunch of plastinated body parts in it. And I was like, oh, that's really cool. You never see a plastinated penis. So I put them in there just to kind of show you a variety of what these might look like. Do what? I'm not sure why the ribbon is there. It didn't say in the book.
To make it more presentable.
Maybe. All right, so the structure of the penis. The portion that you see hanging outside the body is called the body or the shaft, and that's where the erectile tissue is located. On the dorsal side, and I told one group this yesterday, the dorsal side, you have to imagine the penis this way, up against the body like this. Ventral surface, dorsal surface, okay? So on the dorsal surface, you have two corpora cavernosa, so two layers of spongy tissue, and then one ventral on the underside, corpus spongiosum. Corpora is plural, corpus is singular for body, so spongy bodies, cavernous bodies. All right, well, all of these are encased in, again, a tunica albuginea, so that will supply a rigid structure for that erectile tissue to push against rather than just keep stretching. Because what would happen if it kept stretching? You wouldn't really have a hard penis. You just have something that's filled with blood and just kind of like a squishy toy. All right, the corpus spongiosum, that would have been head, extends into the glans penis, so that head of the penis is the glans penis, and that's where a lot of sensory receptors are located. So sexual stimulation occurs there. At the tip, right here, you have the external urethral orifice, and that is the outlet for semen and urine. Prior to being born, when you're born, everybody has foreskin, otherwise known as a prepuce. All right. Common practice used to be that they cut that off. Now there's been a movement to leave it on. So it's a parent's choice on whether or not to do it. But basically what it does is it covers the glans penis and then gets retracted for sexual intercourse and for urination. All right? At the root, so where it's attached to the body, you have two structures that follow the line of the pubic bone. All right? That's called the crura. The crura of the penis. And you're going to see the same thing when we look at the female reproductive organs too. They have very similar structures. And that kind of anchors everything in place. And yes, they are erectile tissue too and do get engorged with blood. All right. That's functional anatomy. How are sperm formed? Spermatogenesis. All right. Again, where this occurs, seminiferous tubules. Where are seminiferous tubules? That was weird. In the testes, right? So, what we have here, imagine this. Red is the seminiferous tubule. This would be one area of the seminiferous tubule, and this would be the lumen. Okay? So, if you notice, there is kind of a division right here. Goes all the way down. So this is one cell. This is another cell. Those gigantic cells that line the seminiferous tubule are called sustentacular cells. All right, they're called sustentacular because they sustain. And if you want to sustain something, what are you doing it? You're feeding it, you're giving it love, you're providing it nutrients, things like that. So these help with sperm development. They support and nourish spermatogenic cells to make sure that they get form into the proper cells. All right, so that's these big ones. Spermatogenic cells, those are all located on the outer surface of the seminiferous tubule. Because like I said, remember, spermatogenesis occurs in this direction. So all of the progenitor cells are on the periphery. Okay, those are called spermatogonia. They are capable of undergoing mitosis. Why do we want to do that? Replenish our population of potential sperm cells. Okay, so these will undergo mitosis and fill in spaces. Meiosis, which would be formation of sperm cells, would go towards the lumen. All right, that's what this says. Mitosis. Those that aren't undergoing mitosis will get bigger, and they'll become a primary spermatocyte. And so that would be these guys here, primary spermatocyte. They're going to stay that way until puberty. So all of this is happening in utero while you're a kid, and it's going to stay there. Things are going to stop and stay like that until puberty. Then after puberty, this whole process is going to keep going until when? The day you die, for males. Okay? The process does slow down over time, and I don't know if you guys have heard this, even though males can technically father children at 99 years old, their sperm end up having problems too. So they can produce chromosomal abnormalities just as well as older women can.
Is it miscarriages more linked to the sperm where like Down sperm is more linked to like women age?
That too. Yep. And that's one of the reasons why you have a miscarriage, because a defect is detected and phew, it stops. Okay. So birth till puberty, you stop there. Once we hit puberty, we start undergoing our meiotic divisions. All right? So primary spermatocyte, first meiotic division. That will give us our secondary spermatocyte. After we make our first meiotic division, we've taken 46 chromosomes, and now we have two sets of 23. Okay, so after that first meiotic division, we have 23. To go from a primary spermatocyte to that takes 25 plus 9, 34 days. Okay? So we're talking 34 days to go halfway through the wall of the seminiferous tubule. After the second meiotic division, we get spermatids. All right? Spermatids still have 23 chromosomes, but we've split those chromosomes from 2 to just 1 chromosome. So we have 4 sets of 23 chromosomes. We split the copies off from each other. All right?
So the two sets of 23, within each 23, there's
A copy.
4 copies?
Yep.
4 sets of 23, there's a half copy?
So at this stage, your chromosomes will look like this. Here, each chromosome will look like this. All right, and notice after that second meiotic division, there's still round cells. So what does that mean? You still have to go under a process of maturation. So the final steps, taking about 21 days to form spermatozoa, you go from a round cell, you develop that tail, you start putting things into compartments, and you make it into a mature sperm that can go and get transported to the epididymis. All right? Are these sperm right here capable of swimming? No. They become capable of swimming once they hit the epididymis. They start swimming once they hit the vas deferens, where the prostate is. All right, you guys are actually going to go over the genetics of that kind of stuff once you get genetics. All right, that's why I'm kind of glossing over some of it. All right, what is the structure of a sperm? We have a head and a tail. Pretty simple. In the head, you have two compartments. This anterior compartment contains something called the acrosome. And in the acrosome, you have digestive enzymes. Okay, what are these digestive enzymes for? Poop, penetrating that egg. In the posterior head, that is where our DNA is kept. Okay, so the DNA that is going to go fertilize and join with the female DNA from the oocyte is in the posterior head. Posterior part of the head. In the neck, you have a lot of mitochondria, and that mitochondria is there to power the tail, which is made up of flagella, which is a flagellum, in fact. Okay, we're not going to go into the cell biology of how flagella works, but again, it's made up of microtubules, and they spin around, and the tail spins around, and it's the sperm swim. All right, I'm not going to show the video, but if you click on this or you go to Canvas, this video, I don't know if it's this video or a similar video, is out there of what sperm look like under a microscope. And it's pretty amazing. You look under there and you're only looking at a fraction of a milliliter under the field of vision. I don't know why it keeps doing that. And there are millions, literally, in that field of view. I'm going to call my son out here for a minute. I showed him a video of what sperm look like under the microscope. He goes, "Those are inside of me? Swimming around?" I said, "Not yet." Not quite yet.
Do you guys know the breakdown of like the fluid in the sperm mounds like it holds like 30% prostate?
No, I'm not going to hold you responsible for the breakdown. It's just for completion purposes. So, sperm is the functional part of semen. What is the rest of semen, or ejaculate? Ejaculate is what's ejaculated out, so this is after it's out of the male body. Of course, we know what seminal vesicles contain. We know what comes from the prostate. And semen, if you want a breakdown of everything, 10% of what's there is sperm. Only 10%. And there's that many swimming around. 30% prostate fluid. Okay, so we need 30% to get those sperms swimming. And 60% comes from our seminal vesicles. All right, average pH about 7.5. Where do you think that pH of 7.5 comes from? What else would you think is in the sperm to make that pH basic? Bicarbonate. Bicarbonate, good. Okay, after ejaculation occurs, Now we're assuming the ejaculation occurs in the female reproductive tract for this, because we're talking about reproduction. In the uterus and uterine tubes, the fluids in there wash away anything that's suppressing sperm activity from the male reproductive tract. So there are inhibitory factors within the semen that are suppressing sperm activity. When the sperm get to the uterus, that's washed away by factors in the uterus. All right? In the cell membrane of the sperm, you have the gradual loss of cholesterol. That makes the cell membrane less rigid. All right, so it becomes more malleable. And why do you want it to become more malleable? One, to release those enzymes from the acrosome once it gets to the egg. And two, so that it can penetrate the egg much easier. All right, three, increased membrane permeability to calcium. All right, calcium is like a second messenger, and that's going to trigger a lot of chemical reactions within the sperm, especially in the flagella, to make them more active. And so they start swimming faster. All right? So a few fun facts about sperm, and I have a really fun fact at the end of this lecture. Take what you want from it. All right, we know that sperm are capable of motility after about a day in the epididymis. Capable. That doesn't mean they're swimming. When they mature after ejaculation, that's when they're capable of fertilizing an ovum. And we talked about the process of capacitation. All right, how much is in each ejaculate? About 2 to 6 milliliters. All right, things that increase semen volume. Abstinence. The longer you go without sex, the larger the volume is going to be. Water. The more water you drink, the more hydrated you are, therefore the more fluid you're going to have everywhere, including semen. Things that decrease fluid volume. You're a newlywed. You have sex like every hour. Like, you're going to have after about 4, 5, or 6 times. I'm kidding, it's not like that, but volume will go down. Also dehydration. If you're dehydrated, you will not produce as much ejaculate. All right? Sperm can be stored up to about a month, and most of that is in the epididymis. All right? If it does not get ejaculated, I talked to some of you about this, what happens to it? Gets recycled. What recycles sperm? Macrophages. So your body literally eats the sperm. All right? It likes, outside of long walks on the beach, alkaline environments, and lower than body temperature temperatures. So if body temperature we assume is 98.6, it likes 96.6. All right? What does it dislike? Vaginas. That's an acidic environment. That's why it comes prepackaged with a raincoat. Let's call that the alkaline fluid, and trail mix. All right. Speed of ejaculation, about 1 to 4 millimeters per minute. And life expectancy after ejaculation, 1 to 2 days. So, if you haven't timed your ovulation properly, you're like, oh, I wasn't ovulating. I just had sex like 2 days ago. And you get pregnant, it's because sperm are still living up in there. Okay, everybody's favorite subject, hormones. I know. Stop your whining. Shut up. Good thing is, all the hormones in the male and female are the same hormones. They just do different things. In the male, you still have LH and FSH, luteinizing hormone and follicle-stimulating hormone. Although, in the male, they have nothing to do with luteinizing or stimulating follicles. Okay? They're still the same hormone, same chemical structure. What they do in the male, luteinizing hormone is from the anterior pituitary. They help secrete testosterone, produce and secrete testosterone. So their target would be the interstitial cells within the seminiferous tubules. Follicle-stimulating hormone, also from the anterior pituitary, stimulates Sertoli cells. And what are those Sertoli cells? Sustentacular cells.
Oh, they sustain, they support and nourish.
They support and nourish the sperm. So if you have follicle stimulating Sertoli cells to support and nourish sperm, they're going to help produce more sperm. So FSH is responsible for sperm production, but indirectly. Testosterone, that results in the growth and division of germinal cells, so that mitosis we see to produce more precursor sperm cells. Estrogens, yes, males, we have estrogens in us as well. They are a secondary product of testosterone. So testosterone's made, then it gets converted to estrogen after FSH stimulates the Sertoli cell. Growth hormone promotes division of sperm. Okay, so notice all of these things that are going for sperm production. And inhibin, they negative feedback on FSH and the anterior pituitary. So they'll shut down FSH and LH, like let's say you're producing too much sperm, for example. Mostly it would be, yes, testosterone can go in feedback as well. So that's just an overview of the hormones. All right, just to give you an overview of what this looks like. So this would be your hypothalamus. This would be your anterior pituitary. So the hypothalamus would secrete something called GnRH, or gonadotropin releasing hormone. That will stimulate the anterior pituitary to release LH and FSH. All right. So during puberty, what happens is hypothalamus starts becoming active, secreting GnRH, secreting LH and FSH. That's going to go down to the testes and start secreting testosterone and start producing more sperm. All right. So that's the basis for puberty. We call this the HPG axis. H meaning hypothalamus, P meaning pituitary, G meaning gonad, because you have an HPG axis in females too. All right, what are the male sex hormones? Again, produced by these interstitial cells. Testosterone is the most abundant androgen. Think of androgens as a catch-all category for all sex hormones. Testosterone is the major androgen in males. All right, when it goes out to the periphery, it's converted to DHT, or dihydrotestosterone, and that is the main testosterone that's going to be responsible for secondary sex characteristics. And again, we know that testosterone secretion is going to begin during fetal development. And what does it do during fetal development? Remind me?
The SRY gene.
Pardon?
SRY gene.
Nope, it comes after the SRY gene.
They drop.
Nope. Well, it does help with dropping too. They're going to help make sure that you produce male genitalia instead of female genitalia. Testosterone. Testosterone. Okay. I will never test you on this. It's just coming to showing you that testosterone, androgens, estrogens, all of those are steroid hormones. All right, and all steroid hormones are derived from this guy right here. Okay. So even though we like to get rid of cholesterol, it is important for producing important things in our body. In the adrenal gland, we can get we can get cholesterol converted to androgens, and then in peripheral tissues, testosterone gets converted into either DHT or estradiol by the enzymes that are within those tissues. Okay? All of this can happen in the testis as well. Testes. All right, like we said, it's a steroid hormone. You have the cyclopentanoperhydrophenanthrene nucleus. You guys ever heard of that before? No? You don't have to know that. I said that for shock value. So steroid hormones. If they are cholesterol, that means they're soluble in what? Fat. They're fat soluble, so they're not easily transported in the plasma, so they have to bind to proteins. Albumin, sex steroid binding globulin, so they're bound by these proteins. Only about 2% of testosterone is actually free. All right, and when they measure testosterone, they measure total testosterone and free testosterone. Okay, we already talked about what it's converted to and where it's converted, what it's converted by. I'm not going to ask you what it's converted by, like the enzyme, but know that enzymes do convert it. So, can you be born without testosterone, or can you be born without the ability to convert to testosterone? Lack of ability, right? Because testosterone, not genetically controlled. Enzymes are genetically controlled. All right? Being a steroid hormone, let's say this is a cell. How does it get inside the cell? By what process? Simple diffusion. Those of you who got that wrong on the test. Simple diffusion. If it's simple diffusion, that means it'll go straight through and its receptor is located either in the cytoplasm or the nucleus. Okay? And what it does is it binds to genes and results in gene transcription, so the production of new proteins. All right? Anything that's left over and doesn't get used goes to the liver and gets converted and gotten rid of. All right, so what does testosterone do? In the fetus, we talked about that at the beginning, okay? And you guys just mentioned that to me a few minutes ago. During puberty, first thing it's going to do is stimulate the enlargement of the testes. You have more testicular tissue, you have more ability to produce hormones. Therefore, you have more ability to produce sex cells, which will lead to the development of secondary sex characteristics after the increase in size of the testes. All right? Testosterone also increases basal metabolic rate. So your cells are more metabolically active, which means once you hit puberty, I had a student in the last cohort said, I was a big kid. And as soon as I hit puberty, like all that weight just went away and I never came back. So you get a lot of energy production as a result of testosterone. Very active body. All right, what are some of these secondary sex characteristics? Enlargement of primary sex characteristics, the penis, the scrotum, testes. All right, so they're already there. That would be a primary sex characteristics. Secondary would be them getting larger. Hair growth. Where's axillary? Armpit, chest, pubic, face, etc. Okay, deepened voice, which means the larynx would be what? Not just enlarged, but thicker, right? So it doesn't vibrate as much. Remember that low frequencies. Increased thickness of skin. And actually, don't even worry about that. They say increased thickness and decreased thickness in women. Nobody's been able to tell me what you mean by thick and thin skin. So we won't worry about that. I lost sleep over that. Testosterone increased muscle development, increased bone density. In fact, it's testosterone that causes a huge increase in muscular development and bone density, not necessarily estrogen. Basal metabolic rate, increased red blood cells to fuel that basal metabolic rate, increased sodium reabsorption. That's a relatively minor effect, but we know it does happen during puberty. And baldness. Anybody know people who started getting bald at like 20? My father-in-law is one of those people. So,
My brother, one freaked out, and the other was like, screw it, shave it.
so my wife, my family's different. We have hair, but my wife said I was starting to thin back here. So I said, you know what? I'm going to start growing my hair out and combing it back. So that's why I have the hairstyle I currently do, because I don't want to get bald. All right, we'll stop right there, and unless you just want to finish this because it's a couple more slides and then go to female. All right, sweet. All right, erection. Yay! We get sexual stimulation, which sometimes could be just sitting there in class and you're like, grade school, junior high. Sexual stimulation increases parasympathetic nerve activity. All right, so again, even though this technically isn't rest and digest, it's driven by parasympathetic. They're going to release nitric oxide. That's going to be the neurotransmitter, and that's going to dilate arteries going to the penis. In particular, these arteries and these arteries. So you have mass blood flow going to the penis. All right? As blood is going to the penis and filling up these spongy tissues, you're going to compress veins. All right? So these veins around the penis are going to get compressed. Less blood's going to go away, so you got a lot of blood coming in, less blood going away. That's going to result in a erect penis. Okay? Okay. So, when the penis is erect, remember, we have sexual activity going on, or math. Yes, I said math. All right. Most of the stimulation comes from the gland's penis. Remember, that has a lot of sensory nerve endings, but psychic stimulation can also play a role. So again, that's at just thinking about things or watching things that you should or should not watch. No, Mom, I swear, it just came up on YouTube. Parasympathetic, again, we talked about the erection, but parasympathetic's also going to cause the bulbourethral glands to start secreting mucus, all right, and that's going to lubricate the penis. Then sympathetic comes in, all right. What sympathetic does is that's going to result in contraction of the vas deferens, all right. So you asked about movement of the vas deferens. This would be happening during sexual activity, but on a basal level, what produces them? Probably parasympathetic, but the mechanism I don't know. Contraction of the prostate, which is going to push fluid into the ejaculatory duct. Contraction of the seminal vesicles, so you're building up you have sperm coming in, you're squirting all this fluid into the ejaculatory duct. Then the process of emission. That's when all of those fluids move into the urethra. All right, so all the fluids are moving into the urethra. Once they're in there, again, what triggers most things in the body? Stretch. So that urethra stretch sends a spinal reflex to the spinal cord, sends signals back to the muscles in the perineal area, and you have ejaculation of that fluid. Okay? And I told them last year, emission. That's when you're at the and ejaculation is okay? to put it to a real life, I try to keep it real with you guys. Hey, next time you are getting it on, you're going to be like, oh, yeah. And they're going to think you're excited. You're going to be like, no, physiology. All right, maybe not, but. All right. The only thing I want to talk about on this slide, after ejaculation occurs, remember, ejaculation is driven by what? Nervous system. No. Sympathetic. So that sympathetic nerve not only is going to result in ejaculation, but it's going to constrict those arteries so you have less blood going in. Less blood going in means more is going to come out despite compressed veins, and the penis is going to return to its flaccid state. Okay? And the whole thing is over. Somebody, and I haven't found the research article yet, clocked an ejaculation occurring at 28 miles an hour. So, faster than a car down a residential street.
What's the video on the bottom right?
The video on the bottom right, no, it's actually not bad. It shows it's a diagram of a penis becoming erect, and then the flow of fluid into the ejaculatory duct, and then ejaculation occurring. So, YouTube views that as a restricted item, so that's why it comes up that way. It's not like Debbie Does Dallas. All right. In case you don't know what that was, that's like 70s porn. All right. Take a 5-minute break.
Question about the vas deferens. It's like sodium dependent, but it's also like norepinephrine dependent, basically.
Oh, the peristaltic waves? Okay.
So it's like even when not having sex,
Sympathetic?
yeah.
Okay.
But then it's not like it's not calcium, it's just
Sodium?
sodium.
Okay. Good to know. Thank you, sir. All right. Females. Good stuff. All right. So, again, some anatomy of this. This is an overview of the anatomy of the female reproductive system. And a good slide to go back to for review. Again, you saw this picture with the males. If you do not have the SRY gene, and therefore you do not have that gonadal ridge that secretes testosterone, by default, you go down this way. Okay? So, if you have a XY male that never maybe let's say that SRY gene is not there, or it never gets activated, you have an XY female. Okay, so that's one mechanism by which you could have, God, what's the word for that? Of course, gender dysphoria, but wrong gender for the chromosome type. All right, major female reproductive organ, ovaries, like the testes for the male reproductive organ. All right, In anatomy, you saw they're held in place by ligaments. You have the main ovarian ligament, and then you have the suspensory ligament, which contains all the nerves and blood vessels. And they are anchored into the pelvic brim. All right, a little bit about the ovaries, but we're going to get an overabundance of this. I promise you. Oh, the handouts that I gave you, they're all pictures that are in here, but those are things that we're going to refer to back to multiple times over and over again. So I thought I'd give you the handouts as well. What are the ovaries made of? Well, we have two parts. We have a thin cortex that goes around all of the outside that has loose connective tissue which holds things together and keeps what we were going to call primordial follicles in place, as well as nerves, blood vessels, and lymphatic vessels. So that's what's going to keep the ovary alive. Then you have the medulla, what's in the middle, and that's going to contain your developing follicles. Okay, and we'll talk about how that process works. We said this with the male. It's covered by the tunica albuginea. And this is what a light microscope looks like. All of this would be the cortex. Here would be the medulla. What else has a cortex and medulla in our body? Kidneys. What else? I'll give you a hint. Yeah, I guess the brain does too. Give you a hint. It's close to the kidney. Adrenal glands. Good. All right, uterine tubes. So, the ovaries are separate from the uterus, but we have uterine tubes that form a pathway from the ovary to the uterus. So, when oocytes get released, they end up going into the uterine tube and traveling down to the uterus. All right. As the uterine tube expands, that's called the infundibulum. And all of these little finger-like projections that are coming off of it are called fimbriae, right? And they're kind of like a basket to collect the oocyte and then draw it toward the uterine tube, toward the uterus. Lining the wall of the uterus. You're going to find out later that the wall of the uterus is kind of bumpy and uneven, but it also has ciliated mucosal epithelium because eggs don't walk. They don't have legs. So they have to have something to transport them, and that's the cilia. Transports it this direction. Okay? Okay. Then we have the uterus. All right, the uterine tubes are directly connected to the uterus. The structure right here. Okay, the only things I want to say about the uterus, notice its location relative to the bladder and relative to the colon. So really, it's between the bladder and the colon. And when there's a baby in there and it occupies all of this space, what's it pushing on all the time? The bladder, right? This is why one time when my wife was pregnant with this guy right here, we went out to get Chinese. She's like, all right, I got to pee before we leave. We peed. 5 minutes down the road, I got to pee really bad. You got to pull over. Like, I had to pull over at a park, and she got out and squatted because she pregnant. Oh, my God. Okay, the job of the uterus is to receive the fertilized egg, which is now called an embryo, because by the time it gets to the uterus, it's about 100 cells. All right? The uterus ends in this structure right here. That's the neck of the uterus, called the cervix. All right? So cervical refers to neck. So you have cervical vertebrae, neck vertebrae. You have a cervix of the uterus, the neck of the uterus. And then the uterine wall. 3 layers, like most of the things in biology. You have the endometrium, which is the inside layer that comes in contact with what would become a baby. So the endometrium. You have the myometrium, which is the bulk of the uterus, this big thick layer. That's smooth muscle, right? Because when you have the baby, you need smooth muscle to squeeze and push. And then the perimetrium, which is a thin layer on the outside. It's made of connective tissue and kind of holds it in place. It's connected to the ligaments of the uterus. And the vagina. All right, so here's the cervix of the uterus, opens into the vaginal canal right here, which then opens into the outside of the body. Okay? Connects the uterus to the vestibule. People see this picture and say, it's a vagina. That's not a vagina. The vagina is in there. Okay? You guys are medical providers. You'll know better. Okay, so this tube right here is the vagina. So it connects to the vestibule. This that we see right here, that's all vestibule. Right? Its job, receive the penis during intercourse. It secretes things, mostly a mucus secretion, and it provides a canal for the fetus at birth. So it's an indoor and an outdoor. Up until either an injury or sexual intercourse, many women have what's called a closing to the vaginal opening called a hymen. It's literally just a sheet of epithelial tissue that's vascularized, so when it breaks, it bleeds. Partially closed. And I found this, just a random Google search. I didn't know this before I looked it up. There's different types of hymen. Okay? So I don't care that you know the names of them, just there is a variety. So maybe you go into pediatrics and you do an exam on a child and you see this and you're like, what the hell is that? Don't ever say that. Okay. Vaginal wall, the inner layer of the mucosa made up of stratified squamous epithelium, a thick layer. Why? Because the vagina takes a beating. That's what it's designed to do. All right? Layer of smooth muscle, then the fibrous coat on the outside. Okay, so very similar structure to the uterus, just not as thick. I always hated this picture because I never saw a picture like it in books, like a male. Like, why do they show, like, it's just open? Like, why does a male have an open thing like that? Because, I don't know. Women. I was thinking of women's rights and all that. But then, if you look in your anatomy books, they do have a picture like this for males, too. So, I was pleased. All right. External genitalia. Labia majora. These are fat folds on either side. Labia minora, right here. Epithelial folds between the labia majora and the vestibule. What does labia mean? Lips. Okay. So when it's in common language called lips, and you're going to have patients say, my lips are hurting. Like, what's wrong with them? No, my lips. They're not actually far off. And you will have patients that talk like that. I promise. The clitoris. This is the clitoris right here. It is a small area of erectile tissue, although there is a wide range of sizes in females as well. Why does it keep doing that? The clitoris is analogous to the penis, so it does undergo erection, and it has crura that go to either side, just like, it's right here, just like the penis of the male, right? It's just smaller in size and does not have a urethra opening. And then we talked about the vestibule. That's what houses everything, including the entrance to the vaginal canal. All right. Down here on the inner surface of the vaginal wall, we have openings to two glands called Bartholin's glands. And these Bartholin's glands, during sexual stimulation, when this erectile tissue fills with blood, they secrete a mucus-like secretion and lubricate the vaginal canal for intercourse. All right, that's it. Simpler. The plumbing in a female is much simpler than the plumbing in a male. So we get through that faster. Then we go into female hormones, which are more important in a female than they are in the male. All right, so this is one of the pictures I handed out to you. All right, I'm going to refer to this picture over and over. There's smaller versions of it on a lot of slides. The first thing you should note if you're going to draw on this picture is you have two major phases of the menstrual cycle. You have the follicular phase and the luteal phase. Okay, the follicular phase and the luteal phase. All right, looking at this picture, the top we have estrogens and progesterones, and down here we have FSH and LH, which are coming from where? The pituitary gland, anterior pituitary to be specific. All right, average cycle is about 28 days, give or take a few days. All right, starting with the follicular phase. Whatever phase we're in, I will try to highlight it in yellow what the phase we're in, so hopefully that helps you guys with understanding where you are in the phase. So when we're talking about the follicular phase, we are talking about everything that's happening right here, okay, in the top half. Now this is just a, what do you want to call it? A diagram outlining the process that happens in the ovary. It's never this neat and clean, okay, so it doesn't like go in this particular pattern. This is just general to teach. Okay, so it starts with a primordial follicle, ends with ovulation, right? So there's your ovulatory line right there. All right, so the process of forming a follicle is called folliculogenesis, right? And it starts with these primordial follicles. Primordial follicles located in this area, so all of these cells right here, not even cells, or follicles, all of those in the cortex are your primordial follicles. And a woman is born with all the follicles she's ever going to have for life. All right? You have 1 to 2 million in each ovary, and once they stop maturing, your reproductive years are over. Okay, so primordial follicle. In each follicle is an oocyte. So these little purple things are oocytes. What is an oocyte? That is the female sex cell that gets fertilized by a sperm. Okay? So the oocyte's surrounded by, in the primordial follicles, a single layer of granulosa cells. So in these larger follicles here, all these individual cells out here, those are granulosa cells. So the primordial follicle, single layer of those. In the primary follicle, which would be this guy right here, primary, notice that you have more layers of granulosa cells, multiple layers, and going from here to here is regulated by FSH and LH. All right, what does FSH stand for? Follicle stimulating hormone. What are these? Follicles. All right, the name matches. Again, in males, not so much. They do have specific names in males, actually. I just don't remember what they are off the top of my head. And clearly I didn't prepare for that in the lecture. Okay, so that's the setup. Next we move to the antral follicle. All right, antral follicles have an antrum, and an antrum is simply a fluid-filled space. All right, so what these granulosa cells start doing is they produce a fluid that goes into this antral space. call it follicular fluid. All right, again, accelerated by FSH and LH, mostly FSH. All right, these green dots here are just to tell you that there's the same green dots that are down here. Those are granulosa cells. Okay, and it's not just one layer thick, as we saw. That's just there for demonstration purposes. All right, when you have the antral follicle, you have a very specific structure. Here you have the oocyte. The green are your granulosa cells. This inner white layer is the theca interna. So it'll be thecal cells. They're epithelial-like, and they secrete sex steroids. So the theca interna secretes our sex steroids, progesterone, estrogen. The outer layer is the theca externa, right? And that's what we call a capsule, and it's vascularized. So that's going to give the follicle its blood supply. I can't see what else it says. Does it? Okay. Oh, so it's connective tissue. Thank you. Am I out of batteries? No.
Wait, what's connective tissue? Both of them, or
Do what?
just one is connective tissue?
The outer one, theca externa. Dang, I'm out of batteries. All right, I'm going to have to do this manual now. Or I'm just stuck. There we go. Okay. So, moving on, then we have our vesicular follicle as opposed to our antral follicle. The vesicular follicle, notice how the shape is changing. Most of it is surrounded by a fluid. What's going to happen here is estrogen from this fluid is going to increase the number of FSH receptors on the granulosa cells. So you're going to increase FSH receptors on those green cells. Okay? That's driven by estrogen. FSH from the pituitary is going to increase the number of LH receptors on these cells. So, from within, you have an increase in FSH receptors. From without, you have an increase in LH receptors. So what does that mean is going to start happening? Those cells are going to become more responsive to FSH and LH. All right? So then LH from the pituitary, as well as the estrogen that's in that follicular fluid, is going to start increasing the proliferation and secretion from both the thecal and granulosa cells. All right? So whatever's being secreted here, whatever's being secreted by the theca interna, go back to previous slides, they're going to be increased. And in this process, you're going to have an increase in size of the ovum, the oocyte, not the follicle, of 1,000 times. So that oocyte's going to get 1,000 times bigger than what it was. Okay. At any one time during a cycle, this is happening in anywhere from 6 to 12 follicles. So 6 to 12 primary follicles are maturing and racing each other, literally, to try to get to a mature follicle. Only one makes it. Okay? Is that always the case? No. Sometimes women release 2 oocytes, and that's how you have dizygotic twins. All right? So that's why I have all these numbered. 9 follicles started this process. There can be only one. You get that reference? Highlander? It's a terrible movie, but it's so great. All right. So, that primary follicle here's how you look at this. That primary follicle is going to start secreting a large amount of estrogen. That estrogen is going to shut down FSH secretion. What do we call that? If a hormone that's coming from one place is shutting down hormones in another place, negative feedback. Okay? So that negative feedback is then going to tell these other follicles who didn't quite get there to involute and die off. All right? So if all this estrogen is decreasing FSH and you're not stimulating follicles anymore, those follicles are going to go away. All right? And then this only this one, number 9 here, is going to end up being a mature follicle that gets to participate in ovulation. Lucky guy. Another name for this is a Graafian follicle. So if you've ever heard this before, and people call it Graafian follicles, it's named after Dr. Graaf. You should be mad. Some dude's naming his.
I was going to mention that.
I'm just kidding. All right. The end of the follicular phase results in ovulation. Okay? So you have this mature follicle. It's full of fluid. It's releasing a lot of estrogen. It's going to respond to something we call the LH surge. If you look at this graph right here, all of a sudden you have this massive increase in luteinizing hormone, and that is the stimulus for ovulation. Okay? When you have that increase in luteinizing hormone, that's going to cause a massive increase in progesterone from the follicle itself. You're going to make a lot of proteolytic enzymes from the theca externa. The follicle is going to start getting a massive, like blood vessels are going to dilate, and you're going to get a lot of blood coming into the follicle, and it's going to get bigger and bigger and bigger. So these enzymes are going to weaken the follicle wall. You have a lot of pressure coming from fluid within the follicle. The follicle is going to swell. It's going to get so big, it bursts. Okay, so literally, the follicle is bursting out of the ovary. Follicle rupture, invagination of ovum, and it's released. That's ovulation. Okay? So, again, LH triggers this, and it's going to produce a large amount of progesterone. It's going to take these actions to produce the ovulation. Okay? This is a summary slide. Again, so I put regulation summary. The cycle takes place from menstruation to your LH surge, ovulation. FSH and LH, as we saw, start the cycle. FSH and LH will increase the number of androgens, estrogen, progesterone, that are released. And those estrogens are going to feed back to the FSH and LH during the first 11 to 12 days to actually increase their production. So that's important. These estrogens will increase FSH and LH once they get to a certain concentration. They're going to increase them. After ovulation, they decrease them. They're inhibitory. Okay? All right. Well, what's happening in that poor little egg while all of this is going on? All right. In the primordial follicles, you have a primordial germ cell. Not even an egg yet. When it goes into the cortex and gets a little bit bigger and gets a layer of follicular cells around it, then it becomes a primordial follicle with a primordial ovum. So that oocyte's getting bigger. This is what's happening in the follicle. This is what's happening to the egg. All right, so we can see like a relationship. When it becomes a primary follicle, you have multiple layers of granulosa cells. You have a primary oocyte. And this is where you stop at birth. Okay, so you have a bunch of primordial follicles. Then you have primary oocytes that are primed and ready to go when the time arises in about, 9 to 16 years nowadays. All right. Now, when this primary oocyte starts to undergo meiosis, again, you're going to separate homologous chromosomes. Chromosome 1 is going to separate from the other chromosome 1. Okay? When the cells separate, so this you'll have 23 once they separate, it's an unequal cell division. So one of these cells is going to give all of its cytoplasm to the other cell. All right? That cell that gets all the cytoplasm becomes a secondary oocyte, and the other one becomes a first polar body, and it degenerates. So what did we lose?
23 chromosomes.
Pardon? 23 chromosomes that would split 2 chances at variability in fertilization. We lost 2 chances right here. Okay? So then it's going to stay that way until it's ovulated and comes in contact with sperm. Only when it comes in contact with sperm is a series of is going to trigger a series of events inside the cell that will cause it to undergo meiosis 2. During meiosis 2, again, you should have 23, 23, 23, and 23. We lost 2 of those, right? You also have an unequal cell division, so only one of them becomes the mature oocyte. The other one is the second polar body that degenerates. So at the end, we only have one oocyte that's capable of getting fertilized. How many do we have at the end of spermatogenesis? 4 sperm from one cell. Okay? That greatly reduces the amount of potential variability you have in children. So the woman determines the baby based on that one oocyte that gets fertilized.
How did the third 23 get dropped? I missed it. So I know that the 2 get dropped since at the beginning of meiosis 1.
Right, so it got dropped here. When this separates, you're going to have 2 23s, right? That gets dropped too. One of those gets dropped too. So in essence, you have 1, 2, 3 that get dropped. All right. After ovulation, notice that it's only the oocyte that gets released with some granulosa cells surrounding it, but the rest of the granulosa cells stay inside the ovary, and they become what's called the corpus luteum. So after ovulation, what happens in the ovary after that becomes the luteal phase. Okay, so you move from the follicular phase to the luteal phase, and that's going to deal with the corpus luteum. Okay, we've already talked about all this. This is just words that explain everything I just talked about already. So another summary slide to go back to. I don't think any of that's new. All right, as far as female sex hormones go, very similar to males. We have the H, P, G axis. We have GnRH from the hypothalamus. We have FSH and LH from the anterior pituitary going to the ovaries. That's going to cause production of androgens, which can go back up and negative feedback, shut off GnRH, shut off LSH and FH. That's a general feedback mechanism. It's not that simple, as we've already seen, but that's a general schematic about how things happen. What these also do, these steroids also do, is they produce secondary sex characteristics. And we'll talk a little bit about that as we go on. So let's continue with the cycle. Right, again, I highlighted in yellow what part of the cycle we're in. Things to notice from that: LH and FSH decrease, progesterone and estrogen increase. Okay, during the luteal phase. So I lost something here. There it is. Okay. So during the luteal phase, what happens? So this is the area we're talking about right here. This is corpus luteum. It's going in this direction. Those leftover granulosa cells and theca interna, they are going to respond to that luteinizing hormone. Okay, the corpus luteum, 80% of it's going to be made up of granulosa cells, 20% is going to be made up of the theca interna. Primarily progesterone is made in the theca interna. Primarily estrogen is made in the granulosa cells, but all the androgens that get produced in the theca interna move over to the granulosa cells and produce even more progesterone. Okay, so in the end, you have a lot of estrogen being produced, but a lot more progesterone being produced, which is why you see this increase in estrogen and a bigger increase in progesterone. All right? When these hormones start increasing, they're going to go back and they're going to inhibit FSH and LH. Lots of estrogen and progesterone inhibits your anterior pituitary hormones. Okay? Essentially, they're going to go down to zero eventually if we let it. So this decrease in FSH and LH will no longer feed those granulosa cells in the theca interna, and that corpus luteum is going to degenerate and become the white body, the corpus albicans. And literally, that's kind of like, think of it as scar tissue in the ovary. And you get enough scar tissue over time, what happens to that ovary's function? Shuts down. That's a proposed mechanism for menopause. Okay. What this is showing you is those theca cells. Remember on the other slide, they were over on this side. Granulosa cells were over on this side. And which way what's being produced where, and what are the products that are going to be produced in the end? Estrogens and progesterone. Okay? I don't care that you know the specifics of the slide. It's just backing up what we said in the previous slide. So I'm not going to go into detail about it. All right. What are the major ovarian hormones? Again, ovarian hormones, not female hormones. So we're not talking about FSH and LH, just estrogen and progesterone. Your principal hormone from estrogen is beta estradiol. Okay, your principal progestin is going to be progesterone. You do produce other types of estrogens and progesterones, but they have effects in the peripheral tissues like DHEA. Remember that for males? Or dihydrotestosterone, DHT. And the rest have weak effects and get cleared by the liver. All right, the liver conjugates them to glucuronic acid, secretes them in the urine. So any extra female sex steroids get secreted in the liver. I mean, in the urine. Jesus. Yes, that's for both. All right. Just to confuse you, there are subphases. All right. So with this first subphase is the pre-ovulatory phase. So notice that starts just before ovulation and finishes just after ovulation. Pre-ovulatory phase. That's about, if you go down 11 to 12 days after the cycle, onset of the cycle as it starts, you have an increase in estrogen. We see that here. Causes positive feedback regulation of luteinizing hormone. So remember, we said prior to ovulation, those estrogens can positively feedback on FSH and LH. So this increase in estrogens is going to positively feedback on LH, and that's why you get the LH surge. All right? The LH surge leads to ovulation. We talked about the mechanism, how that happens with the increase in digestive enzymes and filling with fluid and popping like a balloon. If we don't have this LH surge for whatever reason, let's say you don't have enough estrogen or LH isn't responding like it should, you don't ovulate. If you don't ovulate, you can't produce a child. Okay? So that LH surge is absolutely dependent. Is absolutely necessary, actually, rather. Okay, post-ovulatory phase. Notice that occurs after the pre-ovulatory phase until the end of the cycle. All right, during that time, estrogen and progesterone become inhibitory. So they start inhibiting FSH and LH. All right, as these decrease, you're no longer stimulating the corpus luteum. It begins to involute and turn into the corpus albicans. Okay, then you can start the cycle all over. Does that make sense? Okay, so if you're not stimulating this and it degenerates in the corpus albicans, it's not producing estrogen and progesterone anymore. So the negative feedback on this is relieved, and then you'll start to see FSH and LH increase again to start the next cycle. All right, so that's everything that's happening in the ovary. Now, if you take out this sheet, what's happening in the uterus at the same time? Okay, because we want to produce eggs, but those eggs have to be fertilized and they have to go to a nice fertile garden to sprout up. Okay, so that nice fertile garden is your uterus. Okay, this has 3 phases here, and then they talk about different phases down here. I'm not going to hold you responsible for those things. I want you to know what happens, though. At the beginning, what happens is you have estrogen being released. All right, estrogen driving the process. What estrogen does is it starts to thicken the endometrial wall. So it causes cell division of the endometrial cells and makes it thicker. The progesterone makes the endometrial layer that's becoming thicker more vascular and more glandular. All right, so estrogen makes things thicker. Progesterone makes them more vascular and glandular. So you have a good layer of topsoil, and you've got all the nutrients in it. Make sense? Okay. If the oocyte never gets fertilized, and you stop having the release of estrogen and progesterone to drive this process, they're not stimulated to do this anymore, and blood vessels dry up, they die. If those die, the endometrial tissue starts dying and then sloughs off. Okay? Because estrogen and progesterone dropped and is not making it thicker or vascular or glandular anymore. So you don't get those signals, you drop off. Okay? That results in typically 40 milliliters of blood, 35 milliliters of serous fluid, and a lot of white blood cells. Okay? The white blood cells are there because literally you're ripping away tissue from your body. So white blood cells will prevent infection from happening. Okay? So the picture that I gave you is this picture right here, and it's tying what's happening in the follicle to what's happening in the uterus. Okay, I'm going to go to slide 2 of this, which I think is this slide, the next slide, and the slide after that, and then we'll stop there. Okay? All right. We know that the female reproductive cycle is regulated by hormones. And those hormones start to be released during puberty. During puberty, what you'll start seeing is an increase in gonadotropin-releasing hormone. From where? Where does that come from? Hypothalamus. Right. The trigger for that is the hypothalamus will start increasing electrical activity. And you notice in pulsatile waves. All right? With those pulsatile waves comes secretion of LH. All right? And you notice it's getting bigger each time. So when during puberty, when you have this increase in hypothalamus activity, that's going to drive the increase in hormone release from the anterior pituitary. That's what that graph is showing. All right? During ovulation, during the reproductive cycle, females maintain a certain temperature. At the time of ovulation, temperature will increase by a couple tenths of a degree. Not a whole lot, but just enough for a short period of time. Notice that this is occurring from day 16 maybe to day 24. And if you're taking your temperature every day for, let's say, 2 months, you notice this. This is how my son was born. Like, we track this just so we would know when to practice. So, temperature does increase during this. And that makes sense because where does your temperature regulation come from? Hypothalamus. So if you have increases in electrical activity driving hormones, you're also going to increase temperature. All right. Major hormones. Gonadotropin-releasing hormone, we know what that does. Luteinizing hormone, we talked about what that does. I tell you the slide to go back to go review what that does. So I've included slides to go back to that.